How's it going, folks? Can you guys hear me okay? All right. Just a second, let me send you the link for today's in-lab activity. All right, folks. So I just sent you an email. There are two activities we're going to be doing today. We have two labs we're going to be covering today, labs 12 and 13. Does anybody have any questions, any issues, problems, anything we need to talk about? So guys, after this class today, we only have one more lab next week, Tuesday, and then we have our third exam next week, Thursday, okay? So again, after today, we only have one more lab, which we're gonna cover chapters, labs 14 and 15. And then a week from today, next week, Thursday, we're gonna have our third lab exam, okay? All right, let me share my PowerPoint with you so we can go over the lecture. So we're going to start with the respiratory system. We already started discussing a little bit about it, the respiratory system. Actually, we finished the respiratory system in the lecture. So a lot of the things we're going to go over, they are going to be, you guys already know, it's going to be a review. It's never a bad thing to see it again. And then lab 13 is going to be about the digestive system, okay? The two activities we're going to be doing today, the first activity is going to be using the software, the PhysioEX software. You guys have done this before, you have used it before. I'm going to go over it again so that you know what to do, in which you're going to be running an experiment, doing checking for lung capacity, okay? Like you are walking into a clinic and you are running what we call a spirometer in a patient. Just see the health of that patient's lung. So we're going to do that, and this is going to be for lab 12. And then for lab 13, you guys are going to be watching a video on Edpuzzle, and the video is going to walk you through some important structures and organs of the digestive system. and which you guys are responsible for in the third lab exam. Okay. So we're going to take a look first at the anatomy of the respiratory system, and then I want to spend some time discussing about lung capacity and the spirometer. And again, the spirometer is a test that we do to check lung capacity, to see how healthy your lungs are. Okay? So we have seen this slide before in the lecture, and basically this slide just gives you an overall view of the different structures and the organs of the respiratory system. And remember that our respiratory system is divided into two main apparatus, two main groups of structures. We have what we call the conducting zone, and we have what we call the respiratory zone, okay? Conducting zone is a very appropriate name for the structures because all the structures here that are shown in blue, including the terminal bronchioles, all that they are doing is that they are conducting the air from one point to another. They're passing along the air from one point to another, okay? It's not going to be until we get deep inside of the lungs in what we call the alveoli that we're going to have a gas exchange. So two of the structures here that are shown in orange, they belong to the respiratory zone, and they are the respiratory bronchioles and the alveoli, okay? We're going to go over very briefly through each one of these here and discuss them a little bit more. So let's start with the nose. The nose is the first structure of the conductive zone. And in our nose, we have what we call the nares or the nostrils. And these are the holes that allows the air to go inside the nasal cavity, okay? Now, the nasal cavity itself is the space inside of our nose. And the nasal cavity is lined, the walls of the nasal cavity is lined with a mucous membrane. As a mucous membrane, this membrane produces mucus, the fluid, and the function of the mucus is to moisten the air. As the air enters our nasal cavity, the air is going to be moistened, it's going to be carrying water, and the mucus also is going to warm the air, and the mucus also helps to trap microorganisms, trap fibers and pollutants, okay? We have what is called a nasal septum, which is a wall that separates the two nasal cavity. So this nasal cavity is a bilateral structure. We have a nasal cavity on the right side, we have a nasal cavity on the left side, okay? And in the middle, we have a nasal septum that is both made of a bony tissue on the posterior aspect of the nasal cavity, in the back of the nasal cavity, and in the front, the anterior aspect of the nasal cavity, we have cartilage that divides the two nasal cavities, okay? The bony tissue of the nasal septum is made up of plates of bones that we call the nasal conchae. So each one of these here is a bony plate, and there is a little space in between these bony plates, in between the nasal conchae. And this little space is what they do, the function is that it helps to guide the air from the nasal cavity towards the back of the nasal cavity, what we call the nasopharynx, okay? Let's look now at the pharynx. And it's very important that you guys understand that the pharynx is subdivided into three parts. So if on the exam, I am pointing to any part of the pharynx, you need to tell me if it is the nasopharynx, which is the first part, if it is the oropharynx, which is the section right in the back of the oral cavity, or if it is the laryngopharynx, which is right behind the larynx, okay? Be specific, right? Now, all three of these pharynx here, they belong to the respiratory system. But we're going to see for the digestive system, only the oropharynx and the laryngopharynx are part of the digestive system because we don't eat with our nose, we eat with our mouth. So the food or the drink that we had is going to pass through the oral, the laryngeal, and then go down the esophagus, okay? Distributed in our pharynx, we're going to find a lymphoid tissue, which are the tonsils. So we have the pharyngeal tonsil in the back of the nasal pharynx. We have the palatine tonsil all the way in the back of the oral cavity. And we have tonsils under our tongue, what we call the lingual tonsil, okay? What is the function of the tonsils? The tonsil is a lymphatic tissue, which means that they have immune cells inside. We have B cells, we have T cells, and those cells help to fight infection, okay? A tonsillitis is when we have the inflammation of the tonsils and our tonsils can swell up. Moving on now to the larynx. So we went from the nostrils, through the nasal cavity, through the nasal pharynx, through the oral pharynx, the laryngeal pharynx, and now we're going to enter the larynx. Right? So the larynx is also what we call our voice box, because we're going to find the apparatus within the larynx that allow us to make sound. And the larynx connects the pharynx, more specifically the laryngeal pharynx, to the trachea down below here, okay? There are some important structures of the larynx that you guys do need to know. Among them, we have a big cartilage right here that we call the Adam's apple. This is the thyroid cartilage, okay? And both males and females have them, all of us have them. Right underneath the thyroid cartilage, we're going to have another cartilage right down here. And this one is what we call the cricoid cartilage, which basically it marks the end of the larynx. So the larynx goes from here to here, okay? Below this point here, we're going to have the trachea, right? In between the thyroid cartilage and the cricoid cartilage, we have a little soft membrane right here. We can actually feel it with our fingers. You can stick the finger right there. And this membrane here is what we call the cricothyroid membrane. And this membrane is an important landmark for an emergency procedure that is called cricothyrotomy, okay? And I explain what this cricothyrotomy is in the lecture. So when we are choking and you need to clear your airways, you need to allow air to go into your lungs, in and out of your lungs, then an emergency procedure would be to come in here and make a cut on the cricothyroid membrane, keep it open, put in a tube in there, and then you're going to save this patient's life, allow him to be able to breathe, okay? Another very important structure of the larynx is what we call the epiglottis. And we can see the epiglottis here in this lateral veil. This is the epiglottis right here that I just circled. We can see the little tip of the epiglottis right here. Okay. And what the epiglottis does is that it is a membrane, a mucous membrane that closes the opening to the larynx when we swallow. Every time that we swallow, our tongue will push the epiglottis down. The epiglottis comes down here, closes the opening of the larynx, and it doesn't allow anything to enter the trachea, okay? And it doesn't matter if we're just swallowing saliva. Every time we swallow food, water, saliva, the tongue is going to push the epiglottis down. And we can have a better view of the epiglottis here from this posterior view. So once again, we can see this is an anterior view of the larynx. And you can barely see the cricoid cartilage right there because we have the thyroid gland wrapped around the epiglottis. But we can see right here the cricoid cartilage. But we can see right here the epiglottis from a posterior view. So like I said, every time we swallow, we're going to push this epiglottis down, and the epiglottis is going to close the opening to the larynx. So let's look now at the vocal folds. So the vocal folds are this piece of tissue right here on the edge. And we have two of them, right and left. And these vocal folds, we're going to watch this little video that we watched in the lecture. The vocal folds, they, as the air passes through them, coming down from the lungs and up the trachea, the vocal folds, they are going to vibrate. The vocal folds can change the shape. They can stretch out. They can become thicker. And it's the change in the shape of the vocal folds and the opening and closing of the vocal folds that allow us to make different types of sound. The vocal folds are anchored by this membrane here to the side. And this membrane is what we call the ventricular folds, also called the false vocal cords, okay? And basically what they do is that they just anchor, they support the vocal folds. Let's watch a little bit of that video again, so we can see the vocal folds in action. If my computer allows. Anyway, my computer is really slow lately. All right, since we have already watched that video, I'm just going to move on. You do have access to them on the PowerPoint Maldima, okay? So moving on now to the trachea. And the trachea is also what we call our windpipe. And here we have a cross-section view of the trachea. So this would be the anterior aspect. This is posterior, okay? And we can see here in the front, we have the trachea. This is the lumen of the trachea, which is the space where the air is going to go through. And here in the back, we have the esophagus, and this is the lumen of the esophagus, okay? We can see that the esophagus, then, which is then going to lead inside of the stomach. The walls of the esophagus very muscular. We're going to talk more about it in the next lab 13. And while the walls of the trachea, there are no muscles in them. Instead, we're going to find a hyaline cartilage, which are rings of cartilage that is going to maintain the trachea open, so the trachea doesn't collapse, okay? Now, if we do a little cut on the inner section of the trachea, the inner surface of the trachea, we're going to see that the cells that make up the inner section of the trachea are made of cells that contain cilia. They have cilia on the surface. And remember, the cilia are these little hair-like structures on the surface of the cells. And what the cilia does is that the cilia beats and they push the mucus upward so that we can get rid of a microorganism or particles or pollutants that are trapped in the mucus, okay? This is what we call the ciliary escalator. When we have some type of damage to the cilia of the mucus cells in the trachea, then that individual is going to have more lower respiratory tract infections, okay? We can see that this hyaline cartilage, they have a C-ring structure, and that is because to allow the expansion of the esophagus when we eat something. You know, if you eat a piece of meat or an apple, slice of apple, and it's going to come down the esophagus needs space to expand it and allow the food to go right through it. Moving on now to the bronchi. And once again, I want to show you the different parts. So this all the way up to here, we have the larynx. That again includes the thyroid cartilage and the cricoid cartilage. And then from this point on, we have the trachea. And we can see all the little hyaline cartilage, the rings right here, keeping the trachea open. Okay? Now, this hyaline cartilage, they're going to continue as the trachea now becomes the bronchi. Bronchi is plural, bronchus is singular, okay? So as soon as the trachea splits into two, now we're going to have what we call the primary bronchi. Then this primary bronchi, which is going to bring air inside of the lungs, they will branch off again. Now we're going to have the secondary bronchi. Then the secondary bronchs will also branch off, forming what we call the tertiary bronchi. And that's the end of the bronchi. From the tertiary bronchi, now we're going to have very thin little branches coming off the tertiary bronchi. And these are what we call the bronchioles. And we have two bronchioles. We have terminal bronchioles, which still belongs to the conductive zone. And we have the respiratory bronchioles, to which the alveoli are attached to. And they belong to the respiratory zone. Okay. Looking at our lungs, we can see that the lungs are not symmetrical. In fact, the left lung is smaller than the right lung. The right lung has 3 lobes, superior, middle, and inferior, while the left lung only has 2 lobes. And it's smaller in order to accommodate for the presence of the heart. You know, remember, 2/3 of our heart is on the left side of the thoracic cavity, okay? It's more or less situated right here, right next to the lung, okay? Now, the left lobe, the left lung only has 2 lobes, the superior and the inferior. And each one of these lobes, they're going to be separated by what we call a fissure, which is this line that basically separates the different lobes of the lung. We have an oblique fissure that separates superior from inferior lobe on the left lung, while the oblique fissure on the right lung separates the middle from the inferior lobes. Then we also have on the right lung, we also have a horizontal fissure that separates superior from the middle lobe. We don't find a horizontal fissure in the left lung, okay? All right, so now we are getting deep inside of the lungs, and we can see here the little alveoli, and we can see the bronchioles to which the alveoli is attached to. The first section of bronchioles right here, this is what we call the terminal bronchiole, and then the little branches in which the alveoli is hanging from, is attached to, this is what we call the respiratory bronchioles, okay? Now, can anybody tell me what is the name of the bronchi to which the terminal bronchioles are attached to? So what would this section here be called?
The tertiary one?
This would be the tertiary bronchi, absolutely, okay? So let's do this. Let's take a couple of this alveoli right here. Let's open them up so that we can see inside of them. And we can see what they are made of. So here we have one whole alveoli, and we can see sections of other ones right here. And the alveoli, they are made of 3 main cells. We have epithelial cells that makes the wall of the alveoli. We have macrophages, which are resident immune cells that will trap and destroy any bacteria or viruses or particles that get inside of the lungs, inside of the alveoli. And we also find surfactant secreting cells inside the alveoli, which are the little cells that produce the lipid, the fluid called the surfactant. What does surfactant do? Surfactant decreases the surface tension of water to allow the exchange of gases between the alveolus and the capillary in the lung, okay? Guys, what are you looking at right here in red? Let me show you. There's one over here, another one here. These are showing capillaries that are bringing blood from the heart to the lungs so that the blood can be oxygenated, okay? So as the blood passes here, right next to the alveoli, we're going to have oxygen that is going to go from the alveolus into the capillary. And then the oxygen gas is going to bind to the hemoglobin in the red blood cells. And the CO2 is going to go in the opposite way. It's going to go from the capillaries into the alveoli, so that when we exhale, we blow off that CO2. Does anybody remember what is the mode of transport of oxygen and CO2 in the lungs?
Simple diffusion.
It is transfusion, okay? More specifically, it is simple transfusion. Basically, the oxygen and the CO2, which are gases and they are neutral and they are very small, they can pass directly through the phospholipid bilayer. They don't need a transmembrane protein for that to happen. Okay? Now, I also talked about the Boyle's law in the lecture. And Boyle's law explained the mechanism of breathing. Remember, the Boyle's law states that when the volume of a container decreases, the pressure goes in the opposite direction. That pressure will increase. And the reason for that is because when we decrease the pressure in a container, the little atoms that are inside here in the air, which vibrate, they have kinetic energy, they're going to be finding each other more often. They're going to be hitting against each other and hitting against the wall of the container. And that leads to an increase in the pressure, okay? And this is how our lungs work, with inhalation and exhalation. When we inhale, we increase the volume of the thoracic cavity. We increase the volume of our lungs, and that leads to a decrease in the pressure inside of the lungs, creating what we call negative pressure. What is negative pressure? Negative pressure is vacuum, okay? Vacuum. Now, we can see that this decrease in the pressure inside of the lungs is not by much. It's only 2 millimeters of mercury in relation to the outside pressure, atmospheric pressure, okay? But that is just enough to allow the air to be sucked in our lungs. And that is inhalation. In exhalation, we're going to have the opposite. In exhalation, we are going to decrease the volume of the thoracic cavity, decrease the volume of the lungs, and that will create positive pressure. It will increase the pressure. Not by much, only by 2 millimeters of mercury in comparison with the atmospheric pressure, but that is just enough for the air to be sucked out of the lungs, leading to exhalation, okay? A couple of lung diseases here that I want to talk about. One of them is what we call a group of diseases that we call obstructive lung diseases. And obstructive lung diseases is a condition in which which was going to hinder the person's ability to exhale completely, okay? So they have no real problem inhaling, but when they're going to exhale, they can't fully let the air out. Now, that is different from what we call restrictive lung diseases or restrictive lung conditions. In restrictive lung conditions, the individual is going to have a difficult time fully expanding their lungs when they inhale. They take a deep breath, but it feels like they couldn't fully expand their lungs. A few examples of obstructive and restrictive conditions: COPD, chronic obstructive pulmonary disease, asthma, cystic fibrosis, emphysema. These are all examples of obstructive lung diseases in which you can't fully exhale. Among the restrictive includes sarcoidosis, neuromuscular diseases, pulmonary fibrosis, adastosis, and silicosis. All of those lead to damage in the lung tissue in which the individual cannot fully expand their lungs. So let's talk now a little bit about the spirometry. Like I said, one of the activities you guys are going to be doing today is in relation with spirometry. So you guys need to understand this in order to do the activity and go through the activity, okay? And a spirometry is a test that we can do to measure our lung capacity, to see how healthy our lungs are. It's really unfortunate that we are not in the lab, because we would actually do a spirometry test in the lab in which we can check and see how your lung capacity is, okay? Now, when we do a spirometry test, that is going to produce a little graph, a little result that we call a spirogram. And a spirogram is basically that measurement in which you can look at the values here and see how well your lungs are doing, okay? As I mentioned in the lecture, the way that we do a spirometry test is that you're going to walk into a hospital or a clinic, and then the technician is going to put a tube in your mouth and going to be breathing only through your mouth. And the technician is going to tell you to breathe normally 5 times. You're going to inhale, exhale, inhale, exhale, inhale, exhale, inhale, exhale, inhale, exhale. After the 5th normal breath, you're going to inhale as hard as you can, and then you're going to exhale as hard as you can. And then you're going to, again, normal breathing 5 times, okay? So then the machine, the computer, can measure what is that those peaks, what is the value of those peaks. So the normal breathing, this is what we call a tidal volume, okay? When we breathe normally, like you guys are breathing right now, and the normal tidal volume, an average tidal volume, is about 500 mLs of air that we can take in and out of our lungs, okay? Then when we exhale as hard as we can, and we come in here, and we measure this value right here, this is what we call the inspiratory reserve volume, or the IRV. And on average, the IRV is about 3,100 mLs of air that we can inhale, that we can get inside of our lungs. Then when we exhale as hard as we can, then we get another measurement, this one right here. And this is what we call the expiratory reserve volume, the ERV. And on average, the ERV is about 1,200 mLs of air, okay? Now, when we add all of these up, from here all the way to here, which includes the tidal volume, the inspiratory reserve volume, and the expiratory reserve volume, we get what we call the vital capacity. And the vital capacity, if you add those numbers up, it comes out to about 4,800 mLs. There's just one measurement here that is left. And this is the amount of air that remains in our lungs even after we exhale as hard as we can. That air in our lungs that keeps our alveoli open, keeps them inflated, this is what we call the residual volume. And the residual volume is on average 1,200 mLs of air. When we add the residual volume to the vital capacity, we get what is called total lung capacity, okay? So basically, this is what the doctor is going to be looking at. What is your tidal volume? What is your IRV? What is your ERV? And then it's from those numbers, from those results, that can help the doctor diagnose any type of lung condition, either an obstructive lung disease or a restrictive lung disease. So let's see if we're able to play this video here.
Spirometry is a common test for lung function. It is used to diagnose asthma, COPD, pulmonary fibrosis, and other lung diseases. It can also be a helpful tool to monitor disease progression and evaluate effectiveness of a treatment plan. A tube-like device called a spirometer is used to capture and record air volumes and breathing speed. A spirometry test typically reports 4 respiratory volumes: tidal volume, TV, the amount of air inhaled or exhaled during normal, quiet breathing, without effort; inspiratory reserve volume, IRV, the amount of air that can be inhaled with maximum effort after a quiet inhalation; expiratory reserve volume, ERV, the amount of air that can be exhaled with maximum effort after a quiet exhalation; residual volume, RV, the amount of air remaining in the lungs after a maximum exhalation. These volumes are used to calculate other parameters called respiratory capacities. Inspiratory capacity, IC, the maximum amount of air that can be inhaled after a quiet exhalation. Functional residual capacity, FRC, the amount of air remaining in the lungs after a quiet exhalation. Total lung capacity, TLC, and vital capacity, VC, the amount of air that can be exhaled with maximum effort after a maximum inhalation. This is basically the volume of the deepest breath the lungs can possibly handle, and is an important indicator of pulmonary function, as well as strength of respiratory muscles. Vital capacity can be measured as slow vital capacity during slow, relaxed breathing, or as forced vital capacity, FVC, when the patient is asked to breathe out as hard and fast as possible. While there is little or no difference between these two values in healthy individuals, people with difficulty exhaling usually show significantly lower FVC. Another important parameter obtained during forced spirometry is the forced expiratory volume, FEV1, the amount of air that is exhaled during the first second of forceful exhalation, after a full inhalation. FEV1 is used to calculate the percentage of air that is expelled during the first second. This FEV1 over FVC ratio inversely reflects the resistance to expiratory airflow. In healthy people, it is around 70 to 85%. A smaller number means increased lung resistance. Spirometry is useful in differentiating between restrictive and obstructive pulmonary diseases. Restrictive lung diseases can be inspiratory or expiratory. Inspiratory restrictive are conditions in which lung compliance is reduced, limiting lung expansion when inhaling. This can happen either because the lungs become stiff as a result of scarring or fibrosis within lung tissues, or the respiratory muscles are too weak to inflate the lungs. Expiratory restrictive is when exhalation volume is limited due to weakness of accessory muscles involved in deep exhalation. Restrictive lung diseases are associated with decreased lung volumes, or total lung capacity, TLC. Obstructive lung diseases, such as asthma or COPD, on the other hand, show a normal or somewhat increased total lung capacity, TLC. This is because the obstruction increases lung resistance, making breathing out harder and slower, and this results in increased residual lung volume. Vital capacity remains normal during quiet breathing, but when breathing rapidly, a higher pressure is required to overcome the increased resistance, and forced vital capacity is reduced. A more reliable indicator of obstructive lung disease, however, is the lower percentage of air that is exhaled during the first second of forceful exhalation.
All right. All right, moving on, folks. Let's go over the digestive system now, and then we're going to talk about the activities for today. Same situation. I'm going to walk you through the anatomy of the digestive system, and then we're going to talk a little bit about nutrition. And our digestive system is also what we call our alimentary canal, also called the GI tract or the gut. And the GI stands for gastrointestinal tract. And like the name says, it is a canal, it is a tract, because it is one long tube of about 30 feet long that starts all the way in the mouth and ends up at the anus, okay? Now we also have, besides the organs and the structures of the alimentary canal, we also have what we call the assistant organs of the alimentary canal. And these organs, the food doesn't necessarily pass through them, like it passes through the stomach, like it passes through the esophagus. But these organs, these assistant organs, they are involved with the digestion of the food, okay? They're involved either with mechanical digestion or with the chemical digestion of the food, okay? And those includes the teeth, our tongue, salivary glands, liver, gallbladder, and the pancreas. Most of our digestive system, our alimentary tract, is found within the peritoneal cavity. And remember that the peritoneal cavity, like other cavities in the body, they have a double layered membrane, okay? One section of this membrane is covering, literally wrapping itself around the stomach, and we can see the stomach right here, around the liver, around the large intestine, around the small intestine. And this layer of the membrane is what we call the visceral layer, while the layer of the membrane that is covering the wall of the cavity, we call that the parietal membrane, the parietal peritoneal membrane, okay? A couple of organs, including the pancreas, the kidneys, and the duodenum, they have a retroperitoneal position, and retro means behind the parietal peritoneal layer. Okay? Here we have just an overall view of the digestive system and the alimentary canals, and also some of the assistant organs. So we have our mouth, which is the oral cavity with the tongue and the teeth and the salivary glands. We have the pharynx, and remember, only 2 of the 3 pharynx are involved, are part of the digestive system, only the oral pharynx and the laryngeal pharynx. Then we have the esophagus, stomach, small intestine, and we're going to see we have 3 sections of the small intestine. The first one is what we call the duodenum. Then we're going to have the jejunum, and then we're going to have the ileum. After the ileum, now we're going to enter the large intestine. We're also going to see that the large intestine is subdivided into several different regions. We have the ascending colon, we have the transverse colon, we have the descending colon, sigmoid colon, rectum, and anus. Okay? Let's go over each one of these into more detail. On the roof of our mouth, we're going to find a hard palate and a soft palate. A hard palate is found in the front of the oral cavity and is made up of bony tissue, while on the back of the oral cavity, we're going to find a soft palate. Okay. All the way in the back of the oral cavity, we're going to find this structure right here. This is what we call the uvula, which assists with guiding the food down into the oral pharynx. Our tongue is a very important assistant organ of the digestive system. The tongue helps with speech, but also helps with the digestion, with the mechanical digestion of the food that we eat. Our tongue is held up here in the front by a little mucus membrane that is called the lingual frenulum. And we're going to find a total of 3 salivary glands within the oral cavity. We have the parotid gland all the way in the back of the oral cavity. We have the sublingual gland underneath the tongue. And we also have the submandibular gland underneath the mandible. Now, even though we have these different salivary glands, they all produce exactly the same thing. They all make the same saliva, okay? What do we find in saliva? In saliva, we're going to find is mostly water. We're also going to find an enzyme called the lysozyme, which, by the way, we find this in several different bodily fluids. We find lysozyme in our sweat. We find lysozyme in our tears. We find lysozyme in our saliva. Lysozyme breaks the cell membrane of bacteria, okay? And there's also a very important enzyme within saliva that is called the salivary amylase. We're also going to see another amylase produced by the pancreas called the pancreatic amylase. And a salivary amylases in general, they break down amylose, they break down starch into smaller compounds, which are the oligosaccharides. Looking at our teeth, again, another very important assistant organs of the digestive system. And we have different sets of teeth, depending when you're a baby or an adult. As an adult, we have 32. And you guys need to be able to name teeth. I will ask you questions about naming tooth in the lab exam, okay? So make sure you are familiar with them. Now, the problem here, remember, I'm going to be using a model, a human model, for the lab exams, like we've seen before. So you're going to see the two sets of teeth, the upper and the lower. So when you're naming a tooth, not only need to tell me if it is right or left, but you also need to tell me if it is upper or lower. Okay, you need to have all of that information in there in order to get the full points for the question. Now, it's not too difficult to name teeth. The first two teeth that we see right here in the front, so let's say that we pass this imaginary line right through our teeth. So these first two here are what we call central incisors. So we have right and left central incisors, or it could be right upper or right lower, left upper or left lower central incisors, okay? The next two next to the central incisors are the lateral incisors. Next to the lateral incisors, we're going to find the canines. And then for adult teeth, after the canine, we're going to find the two premolars, also what we call the bicuspid. And these premolars, they are numbered. So this is a premolar number 1, premolar number 2. After the second premolar, we're going to find the molars. Same situation, they are also numbered 1, 2, and 3. With the number 3 molar being what we call the wisdom tooth, okay? As we mentioned in the lecture, our teeth is definitely harder than the bone, since we are constantly applying a lot of friction to our teeth. Moving on, let's take a closer look at the pharynx. And like I said, the pharynx is subdivided into 3 regions, but only 2 of them are part of the digestive system, only the oropharynx and the laryngopharynx, okay? Now we get to the esophagus, and the esophagus is very muscular. We're going to see that the esophagus is made up of 4 layers. So let's do a cut on the esophagus here so that we can see on the inside. So the inner section of the esophagus, the first layer, the innermost layer, is what we call the mucosa layer. And the mucosa layer is made up of epithelial cells. You're going to find the laminal region, and we're going to find a layer of smooth muscle. Okay, that together makes up the mucosa. Does anybody remember, what is the name of the space right here that the food passes right through it? What is the space in an organ, inside of an organ, called? I mentioned this several times before. What are they called? You guys are paying that much attention. It is called the lumen, L-U-M-E-N, a lumen. So we're going to find the lumen in the esophagus. You're going to find the lumen in the stomach. We're going to find the lumen in the small intestine. We're going to find the lumen in the large intestine. The lumen is the space, okay? The next layer of the esophagus is what we call the submucosa. And within the submucosa, we're going to find a lot of glands that produces mucus and enzymes that will continue the chemical digestion of the food that we ate. The next layer is the muscularis externa, which contains two layers of muscle, one longitudinal, one circular layer. And finally, we have a serosa that wraps the whole esophagus together, keep it together, okay? When someone has a heartburn, also called an acid reflux, this is when the contents of the stomach, the acid from the stomach, goes up the esophagus and burns the inner lining of the esophagus. It burns the epithelial tissue. Okay. So now we are looking at the stomach, and the stomach is subdivided into different regions. At the entrance of the stomach right here, folks, we are going to find a sphincter. So we're going to find a sphincter both at the entrance to the stomach as well as the exit of the stomach. The sphincter right here, the entrance to the stomach, in between the esophagus and the stomach, this is what we call the cardiac sphincter. Okay? And remember, sphincter are circular muscles. So the beginning of the stomach, the entrance to the stomach is what we call the cardia. Then we have this upper region of the stomach right here, which is the fundus. Then we have the main body of the stomach. And then we have, right after the body, we have this triangular region here called the pyloric antrum. And then the exit of the stomach, the end of the stomach here, we have a region called the pylorus. Do not confuse the pylorus with the pyloric sphincter. Two different things. A pylorus is a region of the stomach. A pyloric sphincter is a circular muscle at the exit of the stomach, okay? In between the stomach and the duodenum of the small intestine, right? Now, the stomach has two main curvatures. We have a lesser curvature, and we have a greater curvature. Can anybody tell me, within the abdominal pelvic region, and remember the different quadrants, our stomach is where, folks? Is it more on the right side or the left side of the abdominal pelvic region?
The left side.
The left side, absolutely. If you take your left hand and you put it right underneath your rib cage, you are touching your stomach, okay? If you take your right hand and you put it right underneath your rib cage, you're touching your liver. Okay? The inner lining of the stomach, right here, this is what we call the rugae of the mucosa. And inside of our stomach, most of us have a very special bacteria called the Helicobacter pylori. And this Helicobacter pylori, for most of us, it doesn't cause any trouble at all. But for some individual, and we don't fully understand why that is the case, there are some genetic components to it. But for some individuals, the Helicobacter pylori can actually be quite dangerous because it's going to lead to the formation of holes in the inner lining of the stomach. And these holes are what we call ulcers, stomach ulcers, okay? And these ulcers here, they are very painful. These ulcers, they can bleed. And if you have recurrent ulcer, that can lead to stomach cancer, right? Moving on now to the small intestine. And as I mentioned earlier, the small intestine has three separate sections. The first section of the small intestine is what we call the duodenum. And it's very short. It's only about 5% of the small intestine. Nevertheless, even though it's so small, it's a very important part of the small intestine because we're going to have a lot of enzymes coming from the pancreas and also coming from the liver and the gallbladder, which is going to inject those enzymes in the duodenum to continue the process of chemical digestion of the food, okay? So the duodenum is not really involved in absorption, which is the overall most important function of the small intestine, to absorb the nutrients from the food that we ate. But the duodenum is not doing that. Most of the absorption of the food is going to happen at the end of the small intestine in the region that we call the ileum, okay? it's really hard to make a determination. I mean, it's easy to identify the duodenum because the duodenum comes right at the beginning of the small intestine, right after the stomach. But it's not easy to delineate where the jejunum and the ileum starts, okay? So you don't have to worry about knowing where the jejunum is, where the ileum is. Unless I'm pointing here to the beginning of the small intestine. So let's say if I have an arrow on a quiz or exam pointing to any region here, then you don't need to be specific if it is the jejunum or the ileum. You just say small intestine. Let's take a couple of look here at some important assistant organs of the digestive system. One of them is the big old liver, which is one of the largest organs that we have in our body. The function of the liver in the digestive system, okay, is for the synthesis of a fluid called the bile. Now, the liver is going to synthesize the bile, is going to produce the bile, but a lot of this bile, they're going to be stored right here in this little pouch that is called the gallbladder, okay? The gallbladder doesn't make bile. The gallbladder only stores the bile produced by the liver, okay? Now, what does bile do? Bile helps with the breakdown of the fat that we eat. Bile emulsifies fat, it breaks it down, okay? Now, the bile that is produced by the liver and then also released by the gallbladder, this bile is going to enter a duct right here, and this duct is what we call the bile duct. So this section right here is the bile duct. And we can see here that this bile duct eventually is going to inject the bile into the duodenum of the small intestine, okay? Now here in yellow, we can see the pancreas, which is another important digestive assistant organ. And the pancreas produces pancreatic enzymes. And those enzymes, they're going to be collected in this duct right here. And this duct is what we call the main pancreatic duct, which is also going to dump those enzymes inside the little sphincter right here, a little pouch that is called the hepatopancreatic ampulla and sphincter, okay? Then together, the bile and the pancreatic enzymes, they're going to be injected into the duodenum. As the food is passing through here from the stomach into the small intestine, into the duodenum, is going to mix with the bile, is going to mix with the pancreatic enzymes to continue the process of chemical digestion, okay? Moving on now, looking at the large intestine, and we can see here there are several parts of the large intestine, and you guys need to be specific. I will never ask you a question in an exam, either a quiz or a lab exam or a lecture exam, in which the answer is simply the large intestine. If I'm pointing to any part here, you need to be specific to what I'm pointing to. And this is what we're going to go over right now, okay? Now, can anybody tell me, so this here is the end of the small intestine, which is cut it, just a little piece of it. Does anybody remember, what is the name of the final section of the small intestine?
The ileum.
The ileum, absolutely. So what we're looking at right here, we're looking at a little piece of the ileum, okay, of the small intestine. Now, in between the ileum of the small intestine and the beginning of the large intestine, which is this region right here, called the cecum, we are going to find a very important sphincter right there. This sphincter is what we call the ileocecal sphincter. Another name for a sphincter is what we call a valve. So a valve or a sphincter, it means the same thing, okay? Why is it called the ileocecal? It is called the ileocecal because it is between the ileum and the cecum. Okay? And again, the cecum is the beginning of the large intestine. Attached to the cecum, we're going to see this other lymphoid tissue here that we have talked about before. And this is the appendix right there. Okay. All right. So now that the food that got into the large intestine, at this point here, folks, all of the nutrients, they have already been absorbed. So what is the function of the large intestine? The function of the large intestine is basically two. One is for the final absorption of water. Okay, so the leftover water that our body still wants from the food that we ate, the water is going to be absorbed as it passes right here through the large intestine. Two, the large intestine is also very important for the formation of feces. So the feces, they're going to be formed in the large intestine as it moves towards the rectum and the anal canal. Finally, within the large intestine, we're going to find beneficial bacteria that lives in here in the large intestine. It's full of bacteria. And a lot of this bacteria, we want it there. It's not bad bacteria, it's good bacteria. How is it good? It's good because they actually produce different types of enzymes and vitamins, including vitamin D and vitamin K, okay? All right, so let's look at the different parts of the large intestine. The section of the large intestine, folks, that is going up right here. This is what we call the ascending colon. That's the ascending colon. Then this large intestine is going to move from right to left. This is an anterior veil. And this section here, moving all the way from right side of the body to the left side, is what we call the transverse colon. Then the large intestine is going to come down. That is the descending colon. Then the large intestine is going to make this S-shaped curvature right here. That is what we call the sigmoid colon. And the S sigmoid comes from the shape of this section of the large intestine. Then from the sigmoid colon, now we get into the rectum. And the rectum is a section in which the fecal matter is going to accumulate, okay? Then when we have enough fecal matter in the rectum, now the fecal matter is going to pass through the anus. And as it passes through the anus, it's going to pass through two sphincters in the anal canal. We have an internal and an external anal sphincter, okay? These sphincters here are very different. The external anal sphincter is voluntary. You know, when we are potty trained and we learn how to hold on, you know, before we go to the bathroom, that is because we were trained that we know how to control the external anal sphincter. Now, the internal anal sphincter is involuntary. So as the pressure inside of the rectum increases because of the accumulation of fecal matter, there's a lot of nerves that are running right here, embedded right here in the rectum. When the pressure becomes too much, and we all had that feeling before, that's going to send signals to the brain that then relay the signal back down to the internal anal sphincters, telling the internal anal sphincters to relax. And when those internal anal sphincters relax, folks, you better be sitting on the toilet because you got to go, okay? You can't hold that anymore, right? A couple of other structures I want you guys to know. In between the corner of the ascending and the transverse colon, we have this this elbow right here, this corner right here. Oops. What just happened? You guys still hear me okay? Okay, hold on, let me reopen my PowerPoint. All right. So as I was mentioning, at the corner of the ascending colon and the transverse colon, we're going to find this little corner right here, this elbow. This corner is what we call the right colic flexure, also called the hepatic flexure. Can anybody tell me, what does hepatic relate to? What organ in the body? Does anybody know? Hepatic. If you have a hepatic disease, what organ is diseased?
The liver.
The liver, absolutely. So when you are naming this corner here, folks, if I have this question on the exam, you either call it the right colic flexure or you call it the hepatic flexure, okay? It is not the right hepatic flexure because there's no left hepatic flexure, and I mentioned this before with other organs, other structures. So the other corner right here between the transverse colon and the descending colon, this is what we call the left colic flexure, or you simply call it the splenic flexure. Now, this one here is called a hepatic flexure because the liver is sitting right over here. This one here is called the splenic flexure because the spleen is sitting right here. Okay. Two more structures I want you guys to know, actually three more. We can see right here that this large intestine has this very strange look to it, okay? We're gonna see all these little sections of the large intestine. Each one of the sections here, folks, this is what we call a rostrum. So a rostrum are these little sections of the large intestine. And this rostrum is formed because there is a line of muscle, smooth muscle right here. You see this line right here? This line is representing a muscle that is called the teniae coli. And this teniae coli gives this semi-contracted look of the large intestine, okay? And finally, here in yellow, we see a membrane that is holding the large intestine in place. And this membrane is what we call the mesentery. Again, the mesentery is the membrane that holds the large intestine to the back of the abdominal pelvic region. Okay. Let's look at this little animation here as the food goes down through the alimentary canal.
Here we'll show what happens when we digest food. As food enters the mouth, the teeth grind it down during chewing. This is called mastication. Salivary glands release saliva that moistens the food and helps to break down sugars. The tongue positions the food so it can be swallowed. The food moves through the throat and into the esophagus. Food moves.
Let me show you right here. It's a good view. So here we have the epiglottis, right in the back of the tongue. And this here is the vocal folds and the opening to the trachea down below. Okay, so that would lead into the lungs. And here is the esophagus, which is going to lead into the stomach.
Food moves down the esophagus by peristalsis. These are wave-like contractions of the muscles in the digestive pathway that push the food along. Next, the food enters the stomach.
I want to stop right here. So now the food just passed through the esophagus and now entered the stomach. And we can see right here, this is a sphincter. Does anybody remember? What is the name of the sphincter at the entrance to the stomach?
The cardiac sphincter.
The cardiac sphincter, absolutely. And you can see how the sphincter, contracts and relaxes to allow food to pass right through it.
Where it's showered in gastric juices and proteins are digested. The food then passes through into the small intestine.
And right here, we can see another sphincter. What is the name of that one? At the exit of the stomach.
The pyloric sphincter?
The pyloric sphincter, absolutely.
Here the food is showered with bile that digests fats and pancreatic juices that contain enzymes to digest carbohydrates, fats and proteins. The walls of the small intestine also release enzymes. The small intestine is over 5 meters long and the food moves along by peristalsis, as in the esophagus. After the food leaves the small intestine, almost all the nutrients have been absorbed. Next, the food moves into the large intestine.
Right here, we can see the connection between the end of the small intestine, the ileum, and the beginning of the large intestine, the cecum. And there's another sphincter right there. Does anybody remember the name of that sphincter? Between the ileum and the cecum. That's the ileocecal sphincter or the ileocecal valve.
The main role of the large intestine is to absorb water, nutrients and salts and convert waste products into a more solid form that can be excreted from the body as feces. The colon ends in the rectum and anal canal. By this time, water has been absorbed and the feces are ready to be expelled from the body through the anus.
A cool little animation. All right, the very last thing I want to talk about, a little bit about nutrition. We all have seen this slide before, this drawing before, the little food pyramid. And yes, it is that important to have a balanced meal that contains different grains, vegetables, fruits, milk, meats, and beans. Now, why do we eat? Do we just eat because we like the taste of food? No. Yes, it's a bonus. It's a good thing. We evolved in order to like food, have pleasure in the taste of food, because food is that important to us. I mean, think about it. If none of us, if we didn't evolve during the process of evolution, if we didn't like the taste of food, we would probably have died out as a species. Okay? But over time, we evolved and there was this connection between our taste buds and our brain that when we eat something that is pleasurable, actually releases endorphins from our brain that gives a pleasurable sensation. This is how we evolved over time. Now, Of course, that is so that we can get the nutrients from the food that we eat, so that we can get the building blocks for our cells, so we can get the lipids, so we can get the amino acids, that we can get the carbohydrates, the glucose, that we can get the vitamins from the food that we eat, okay? And each one of these vitamins, they play essential role in the function of our cells and in the function of our organs. You know, we talked about, for example, vitamin A before, and we have seen how important the vitamin A is, especially for our night vision, because without vitamin A, we don't have the protein that allow us to be able to see at night under low light, okay? Vitamin B is extremely important for the production of red blood cells. Vitamin C is a very important antioxidant. Vitamin D is very important for our absorption of calcium, and also very important for our blood clotting. We also talked about that before. So you can see that every vitamin plays an essential role in our cells, in our body, okay? Now, that being said, too much vitamin can be dangerous. There is such a thing as called hypervitaminosis, which is the overconsumption of vitamin. Just to give you an example, too much vitamin A, for example, has been linked to osteoporosis, has been linked to bone loss. And especially the elderly, you need to be careful with the vitamin A, because the elderly in general, especially older women, after menopause, they have more propensity for osteoporosis, for bone loss. And if you're over intaking vitamin A, that is going to exacerbate that osteoporosis, okay? So you still should stay within the recommended amount of vitamin. You know, if you take a vitamin supplement and in the bottle there it says, take 2 pills a day, then you think to yourself, okay, if 2 pills is good, I'm going to take 20, and they're going to make me Superman. No, all that's going to happen is that you are going to over consume vitamins, and that is going to compromise some of the functions of your body. Okay. Any questions, guys? All right. So let's talk about the activities for today for the labs. Like I said, I sent you an email earlier in the class. One of those links is for a virtual lab that is going to talk about the respiratory system and go over spirometry. And the other link is for the Edpuzzle video that is going to walk you through some of the organs and structures of the digestive system, okay? Let me show you what the virtual lab activity is going to look like and what you need to do. So if you click on the link that I sent you, this is what it's going to take you to. Again, we have done this before. You are familiar with this. So make sure that you read the objectives of the experiment, and then you're going to hit next. Again, you're going to have an introduction here that describes the mechanism of breathing, inhalation, exhalation. What are the muscles involved with breathing? Make sure you take a good look at them. And this is going to be also very important. More information for you to help you prepare for the exam. It's going to describe the different values of a lung capacity. What is the tidal volume? What is an IRV? What is an ERV? Then you're going to have a little pre-lab quiz. This is not our in-lab quiz. You can just answer them. These are not graded. This is just to see what you know before you do the lab. And then you're going to get to the actual experiment, okay? Like I said before, every step along the way, that's going to be instructions on what you need to do. Read those instructions carefully because otherwise you're not going to be able to move through to the next step, to the next phase. Make sure you read them, and then you start doing the experiment, okay? You're going to do the experiment all the way at the end. You're going to collect some of the data, record some of the data. Then you're going to have a post-lab quiz. Also, you're going to answer them and submit. You don't need to do the summary at the end. So you can take the quiz, but you don't have to do the summary. Keep going all the way to the end. At the end, you're going to have the option of saving this as a PDF file. Save the experiment as a PDF file, and you're going to email the PDF file to me. This is going to account for your in-lab participation today for the Lab 12, for the respiratory system. Okay? After you're done with this, you're going to start doing this one first. After you're done with it and you emailed me the lab report, the PDF file, then you're going to go on Add Puzzle, and you're going to watch the video, answer the questions. You don't have to submit anything for the Add Puzzle. I can track to see who has done that. After you finish with the Add Puzzle, then you're going to jump on Lima, and you're going to take the quizzes for today. There are 2 quizzes already open. Quiz for Lab 12 and the quiz for Lab 13. Make sure you submit the quiz at the end after you completed the quiz. And after you're done with both of the quizzes, you're good to go. Okay? Any questions, guys?
I have a question about the digestive system.
Uh-huh.
So for the digestive system, when someone gets obese, does their intestines and everything grow with them, or does it stay the same?
I mean, expands with them? Oh, absolutely. Absolutely. So there's an enlargement of the different organs. There's an enlargement of the stomach. There's an enlargement of the small and large intestine as well. Yeah. It's just because of to accommodate the excess food that they are intaking. Any other questions, guys? All right, so let's get started.